We have Dr. Andreas having us today with a talk on drug discovery and AI. This is a very intriguing topic, even if you don't know or have any background under this. I'm not going to do a huge introduction. The guy has a Wikipedia page. I think that's enough. Right. Thank you, Doctor.
Very good. Thanks for welcoming me here. You can hear me okay? Yeah? Is it online okay as well? Yeah. Otherwise, let me know. So I'm actually a life scientist, and during COVID, I think it happened that I was thinking about what we are trying to do in drug discovery with data and machine learning models. And I was thinking about which data do we have? Does that actually align with what we want to achieve? And well, the answer is sometimes yes, sometimes no. And this talk is meant to be a bit of a reflection on what we try to do in the life sciences in particular, what's particular about the data in that field. It's different from many other areas. The data we have is very conditional. What we want to predict is actually human outcomes in drug discovery specifically, where we have little data, it's very conditional data, it's tricky data. And so here I want to outline problems with the data that we have, as well as some areas where we can, in my opinion, actually constructively use data in the area of drug discovery. And so to just tell a few words about myself as well. So I come from chemistry, and it's still an important area for me. That's the agent we can put into a living system. But there's also the area that we use chemistry in. So that's more biological, it's the medical area. And ML, AI, I see that mostly as a tool. So it's not a means to an end. Right now, there's quite a lot of tech push, right? It's about fancy networks and so on. For me, the question is more which data and which machine learning model should we use and put it into a process to improve things, okay? So I'm very much here at this interface in the middle. Past, I was actually in very different organizations. So the last 15 years, I was in Cambridge in Britain. We also started some companies. I will come talk about those companies as well. So Helix, Farminable, and Pangea Bio. Most are in Europe, but Pangea Bio is also here in Hub 71, locally in Abu Dhabi. And I was working for Big Pharma as well. And that's what I was always trying to tell my students. Yeah, work in different organizations in different countries. You learn something very different, and all of that matters. So I like to translate things to practice. But academia is actually a good place for that, because you can have many different partners. You can start your companies and put things into practice. So I don't only want to train models. I want to put that into practice to improve drug discovery. That's basically what we're trying to do. And so here, so we started a few companies, as I said, but any statements I will make here, they're in my capacity as an academic. So it's a reflection on data in the life sciences. How can we use that in practice? And there are actually 2 articles we wrote in Drug Discovery Today in 2021. There's hopefully a new article coming up in Nature Reviews Drug Discovery on a few topics I mentioned here as well. What is realistic? What are illusions? Sometimes you have models, you have machine learning approaches that are quite difficult to translate into practice. You use them for decision making in a pharma company. Many of you will have heard of that, this disconnect between machine learning models and implementation and practice. Sometimes it's claimed that pharma is slow in adopting AI. Well, there are good reasons for that. The data looks very different. The questions we want to answer, sometimes a machine learning model doesn't translate to a better answer in practice. And that's exactly what I will explain in the talk today. And is also what is described in those 2 articles. So in case you're interested in reading more, please go to those Drug Discovery Today articles. They are written in quite an understandable, generally understandable way, whether you have a life sciences or computer science background, and tells you more about biological data, what is particular about that. It's important to understand the field where you want to apply an algorithm before building a fancy model, okay? Otherwise, you only have a fancy model, but you still cannot solve any practical problems. So this is what I will talk about. So the prologue, so to speak, is very brief. It's only 1 slide. Find out what matters in what you want to do with machine learning. And for drug discovery, that is the clinic. It's not a fancy model. It's not optimizing precision on some proxy metric. It's putting the right compound into the right patient. That is drug discovery. Also applies to other areas. Think about the end goal of what you want to do. So there's only 1 slide. Then I will talk about the current state of AI and drug discovery, review that a bit. I'll tell about the data and model process disconnect, and that's quite big in the life sciences. And the bottom message of that is make sure that your use case, the data you have, the model and the validation and the process are aligned. It's quite easy to find a solution for the wrong problem, but that actually doesn't advance anything. And I will tell a bit and share about my experience working in big pharma, academia and startups, trying to put that into to good use, into practice. Okay, and we also, of course, now we're in Abu Dhabi here, and so we're trying to build an ecosystem here in drug discovery, where, of course, computational modeling, bioinformatics, and so on is a strong component. There was the Emirati Genome Program, and so on, and I will briefly have a few slides on that as well. So what matters? So this is from the article I just mentioned. So this is a numerical simulation we've done, and what we tried to do is model the impact of being faster, cheaper, and better in the different phases of drug discovery. So what you see here is in the black color is making better decisions, putting a better compound into the clinic, reducing failure rates in the different phases of drug discovery. White is being faster, and that matters in drug discovery because if you're faster, you have longer patent life. You usually have, for small molecules, 20 years patent life. Yeah, and when you have exclusivity, you have higher margins, so you have higher return. So speed matters. And we've modeled the impact of cost being cheaper in different phases of drug discovery. And we've done that across the different phases of drug discovery, from finding a compound active against a protein, optimizing that compound, to the clinical phases. And so clinical phase 1 is a safety phase. You put your compound into healthy patients, and if nothing happens, you're actually quite happy. It's safe enough. Clinical phase 2 and 3 are efficacy phases. So you treat diseased patients, and you need to have an activity of the compound to cure the disease. So it's very different, clinical phase 1 and clinical phase 2. And clinical phase 3 is confirmatory efficacy, as it's called. So you put your compound into a larger patient cohort to confirm that the compound really works. And so what you see here is the highest bars, and we modeled all of the effects. We put that onto a monetary axis, because if you're faster or cheaper, you save money, so you have a better return on investment, basically, in those cases. So the higher the bars, the better. And so we mapped that into one axis, and you see the highest bars, actually the black bars here in the clinical phases. But if we reduce failure rates in clinical phases, that has the biggest impact on a financial project success, which matters, of course, in pharma, because money is not infinite, yeah, and you need to bring drugs to the market that succeed. And so why is that? Those phases are long, costly, and failure rates are high. Okay, so the clinic is what matters in drug discovery. And there's a slight disconnect. So this is what matters: bringing the right drug into the right patient, dosed in the right way as well. You have different administration routes and so on. So this is what matters. The clinic is what matters in drug discovery. So the question is, how can we support drug discovery and clinical decision making using data? And one way is, for example, biomarkers. If you have biomarkers, so you stratify patients, in cancer, for example, you triple clinical success rates. You go from 3 to 8%. It's a huge effect. It's far larger than the failure rate reduced by 20%. But what happens in many cases, and especially the first generation of AI and drug discovery startups, some of which actually were not so successful recently, they focused on the earlier phases of drug discovery, okay? Finding a compound that's active against the protein and so on. Okay, so what's the bottom line? Pick what you focus on. Focus on what matters in a given context, what matters in drug discovery, decisions that are the right decisions that matter in the clinic. A compound that is safe, a compound that's efficacious, so that cures the disease. Okay, so that is what matters in our area. Okay, let's review what actually happened. Computers in drug discovery is nothing that novel. There were, in my opinion, 3 waves so far. One was in the 80s. Here's a Fortune cover from 1981. Proclaiming the next industrial revolution. There was actually an interesting year. Silicon Graphics was founded. So some of you who have been in computer science for a while might remember those machines, the Octane, O2, and so on. So these were the first machines that could do proper computer graphics, as you can see here. And so that was a year when DOC, so it's one of the docking algorithms, where you can have a protein structure, put in a ligand, was published, and so on. That was the first wave, very graphics driven, very structure driven. There was another wave around 2000. The Human Genome Project, for example, was published. The draft version of the Human Genome was published in 2000 as well. There was a biotech bubble in 1999-2000. That was very much bioinformatics, very data-driven, very different from the wave in the 80s. And today, these are some of the headlines you find in the newspapers and so on, how AI is changing drug discovery, et cetera, et cetera. We will come back to the numbers if that is really the case. Discovering a drug means you get approval. The drug is safe and efficacious, okay, and it's approved. You have clinical success. The numbers don't quite tell that. So it reads quite similar, right? In the 80s and currently, but this 40 years apart. Did nothing change? Of course not. We have GPUs, we have TPUs, we have various convolutional and so on networks, right? Software and hardware is much more powerful. What pharma also did is they annotated their data with metadata much better than before. In the old times, companies tested many compounds against a particular target, stored the data somewhere, but metadata annotations were very poor. So there was not even a standard identifier for the protein target that was screened against, and so on. I was at Novartis as a postdoc, 2007, 2008, and they cleaned up their data in that time. And actually, knowing what you know, I think that's already half of the success story of AI, because you're able to search data. You know what you know, okay? And that is already a crucial step forward. So powerful hardware, powerful software, better metadata, we can generate much more data. But what did not really change, unfortunately, is in many cases we don't understand disease well enough, and we don't have what's called early stage assays to be predictive for the in vivo situation. So knowing what to test in the test tube in order to go into patients. Think about Alzheimer's, for example, disease mechanisms very poorly understood. Think about many other diseases, we don't really have enough understanding of proper endotypes of diseases and so on, okay? So we don't really understand what matters for the in vivo situation. So we can generate lots of data. Is that data relevant for decision making? It depends. It's a needle in a haystack problem, okay? And so I think many things did change, hardware, software, algorithms, and so on. Metadata changed. But what did not change as much is disease understanding and knowing which early stage assay is predictive of human systems. And that's a problem because you can generate lots of data, but the data is very high dimensional. And if you don't have enough data points, even if you do feature selection or fancy model generation, you have a very, very high dimensional problem. You don't have enough rows in your system, so you still don't know what to do with that problem, okay? So if you have disease understanding, you can you have a much smaller hypothesis space to test, basically, statistically speaking, right? And you see much more in your data. Quite often, it's large-scale data generation. So my bottom line is, whenever you generate data, hypothesis-driven research is better than hypothesis-free research, okay? That only generates very high-dimensional data sets, okay? But that is a summary of what's similar, what's different between those times. To put things into context, for many of you, that will be the first wave going through one of the, I think, bubbles. For me, it's the second one, and it was very enlightening to go through that the first time. When the biotech bubble 99, 2000 was around, or the human genome sequence was published. So these were some of the expectations the year after. The number of drug targets will increase by at least 1 order of magnitude. Target validation will become a throughput process. There will be more drug targets, 3,000 or 10,000, compared with, at the time, 483. Well, it didn't quite happen. So more recent numbers, and that's the most recent numbers I found. Maybe the number is 800 now, but it's maybe 800 or so proteins that are targeted by current drugs, and certainly not 3,000 or 10,000, okay? So it's very easy to overestimate what will happen. On the other hand, the human genome allowed us to at least have a map of our building blocks. It's quite a static map, okay? It's not a dynamic one. What we would like to understand is what are interactions in a given cell type, in a tissue, at a given time, in a diseased patient, and so on, okay? That's what you would like to know. That's a dynamic model. But at least we have a static map of our biological building blocks. We didn't have that before either. There were estimates humans have 200,000 genes or so, right? So the Human Genome Project told us it's about 20,000 or so, somewhere in that order. Although there are now discussions again about the dark genome and so on, so who knows how those numbers develop. But the point is, it's very easy to overestimate at a certain point in time what will happen in the near future if you translate things to practice. And that's what interests me. How can we use technology and potential and translate that to applications? So not just a fancy paper with a new method, a better decision and picking the right compound to put into a patient, okay? Because that's what matters in the end. So what was happening financially? So there was quite a lot of funding going into AI and drug discovery. So it's about 16 billion or so until 2021. Difficult to get those numbers because you don't quite know. In big pharma companies, it's difficult to really get precise numbers together. But it's certainly a lot of money. Okay, there was a lot of focus on that. What did we get for that? So that's from a Nature Reviews Drug Discovery paper, 2022. And I can really recommend that paper. So what they did here is they compared projects in AI first companies and big pharma. And what you see here is the projects in different phases. Discovery, so that's test tube or animal stage, and phases 1, 2, and well, 3 doesn't really exist here, that's the point. And in big pharma, the distribution looks quite different. It's quite a different scale, right? Big pharma is much bigger. But what you see is you have a very different distribution across clinical phases. The real proof point for drug discovery comes in phase 2. You put the compound into a patient, diseased patient, and you cure disease, or you see an improvement, at least of that state. And you see here in big pharma, you have compounds, a few hundred or so, in phase 3 clinical trials. However, in AI native companies, that number is actually very small to non-existent. It changed a little bit in recent times. You have some compounds in combined phase 2, phase 3 trials, and so on. But not on the micro level, so the individual company level, as well as the FDA, so that's the regulatory authority in the US that approve drugs. The numbers didn't really change, not on the micro level, on the individual company level, also not on the macro level, okay? So right now, and you might remember those headlines I've shown before, how AI is changing drug discovery. Well, drugs means putting compounds into patients and they work, okay? And if you look into those plots, you see that to a good extent, that did not really happen yet, okay? And the reason is that life science data is very, very tricky. On the other hand, computational methods have been used in drug discovery for decades, okay? So that there has always been a contribution of computational methods. Those docking methods I mentioned, we have a ligand, like you might have heard of AlphaFold, you can predict a protein structure, dock a ligand. This type of methods, not AlphaFold, but the docking methods have been used for 30 years or so in pharma, okay? It's just difficult to quantify that contribution. Sometimes those headlines that we read in newspapers, they are a bit over-optimistic in my opinion. Funnily enough, after in 2018, AI changed drug discovery already. You see much more careful headlines in recent years. So that's from Nature Medicine, inside the nascent industry of AI-designed drugs. And this is what some of the CEOs of such companies, AI and drug discovery companies, then say. There's no shortcuts to drug discovery. The trials are still in the early days. Yeah? That's 5 years after the big headlines we've seen. But in some sense, we are actually still quite early, and I agree with that. Yeah? It takes quite a while until you figure out which methods do really work, until you change a big organization. That all still takes time. Yeah. So we had 10 or 15 or 20 years, but in one sense, it's still early. I agree with that. But you need to put things into practice. That is important. Okay, so conclusion, neither on the micro level nor on the macro level, AI really shifted the needle yet in drug discovery. Biomarkers is one of the big success stories though, stratifying patients where you understand enough of disease, 3-fold higher success rates in clinical trials. There's lots of activity in early stages, but it's often, and that's in this Nature Reviews drug discovery paper, explore targets, close analogs. So it's just redoing things that people have done before. And there's a good reason, because if you build a machine learning model, you rely on data, and your model is more confident in areas that you visited before, where you have data points, right? Extrapolation out of domain prediction is very, very difficult. True everywhere, also true in biology. So the question is, where's the novelty here? And if you read newspapers and headlines, I actually switched to thinking, do I read about inputs or funding successes or output? Okay, so of course it's a success if you raise $100 million or so for a company, very good. But then you need to transform that into output. Okay, success in a clinical trial. Okay, always see, do I read about input or output? And it's always important to compare to the null hypothesis. So the first AI designed drug will be celebrated by the media. Happened many times already. I mean, there were 10 or so first AI designed drugs in the media already. But the question is then, what is your null hypothesis? If you put tens of billions of funding into something and it costs you 1 billion, difficult to quantify, but let's say 1 billion to discover a drug, even your null hypothesis would give you 10 drugs, right? And so always compare to that as well. So that's the first part. That's where we stand. I find it like technology. It's excellent to see that. It's important to put money onto that so we can actually try new things. That's very good. But it's also important to not get stuck in that tech fascination phase, I think, and to transform that into real applications and to use models. And that is very, very tricky, but it's very, very important because that's where real change comes from. And I will tell you about why life science data is actually tricky. So the short summary of the next few slides is technology is fascinating, high throughput data generation is fascinating, but using it productively in a given use case is actually quite difficult. And I will point out 3 main reasons in the life sciences for that. Life science data is very difficult to label, and if you build a machine learning model, a supervised model, but in some sense also an unsupervised model, because if you cluster your data, at some stage, if you make a decision, you always need to have a threshold. So at some stage, you always need to come to quantitative or qualitative labels. So it applies in any practical decision-making case. Life science data is difficult to label, and hence training meaningful models is difficult. A model is only a model. You need to embed that into a context, which is a process, a use context. And validation relevant to the use case is very, very difficult, even impossible in a way. I was talking to a few people in pharma companies and asked them at AstraZeneca or so, some of the big companies, so how do you validate models? And they just said, well, what gets used in the end, that's validation, yeah, because you use things in many different contexts and you have nearly a gut feeling. It's not a numerical quantitative evaluation in some cases, yeah. At some stage you make a decision because you cannot have enough prospective use cases in practice. You cannot put every compound into the clinic. It doesn't work to validate your model, yeah. So you always operate in some proxy space for model validation. I will outline that. Why is it difficult to label data? So sometimes if you read a machine learning journal, you find a paper that tries to address this question: does a certain drug cause an adverse drug reaction, yes or no? So binary classifier, 99% average success in classification, average accuracy or so. Paper gets published, people would think, okay, we have a pretty good model, right? Well, in practice, no. And why not? Because in practice, that all depends, okay? Do I see a certain adverse drug reaction? Depends on the genotype of the patient. What's the disease endotype, right? In asthma, diabetes, and so on, there are different endotypes, subtypes of the disease. Then you take a certain dose of a drug. So effects are dose dependent, it's quantitative. Then we might see an adverse drug reaction, which comes in an arbitrary ontology, okay? Phenotypes are very difficult to categorize, yeah? So then we see an adverse drug reaction, but only in a certain fraction of cases, 12% of cases or whatever, with a different severity. And maybe you only see it if you co-administer your drug with a different drug from a certain class or particular drug. And maybe you see that only in males or females, maybe only after 3 months of dosing and so on. That's what things look like in reality in the life sciences, okay? Things really depend. And so does a certain drug cause an adverse event? Well, it depends, right? Even water can be toxic. Okay, if you drink 3 liters of water in half an hour, you dilute your electrolytes. Can be a problem if you're on a marathon and you only drink water. You shouldn't do that. So you dilute your electrolytes, right? This is death by water intoxication. That can be a problem. So things depend on dose, right? Of course, water is not toxic, but you have many other molecules that are toxic at other levels. Maybe a few grams. Is that then toxic if you build your model? It depends. Depends on the use context, right? So labeling your data can be very, very tricky. Another example of that, ketamine. So it's a compound. It's quite an interesting compound. And I want to illustrate why it's actually difficult to label biological and medical data. So that's been used, that compound has been around for ages. It's been used as an anesthetic originally at a certain dose, IV, so intravenously, 2 milligram per kilogram dose. Has been approved very long time, used in animals and so on as well. But it's also used as a street drug. In 2000, the effect as an antidepressant was discovered, but dosed lower at only a quarter of the dose. It's also used as a bronchodilator, so it has an effect on your lungs. And that is not exhaustive, right? I mean, if we test the compound in different use contexts, we get a new label, right? So we don't know all those labels. And the labels depend on dose. Yeah, if we dose higher as an anesthetic, if we dose lower, then it's an antidepressant, okay? So things depend, as shown before. It's been thought to act via blocking a particular receptor, NMDA receptor it's called, but other NMDA blockers do not work, so that cannot be the whole explanation. Again, we don't know all the labels. And also the opioid system is implicated in the action. If you give antagonists of the opioid receptor, you see a different effect, okay? So, but no one quite understands why. And in animal models, a metabolite, so not this compound, but a product that the body makes out of this compound, has been found to be active in animal models, but not in humans. Okay, the details of this compound don't quite matter. My point is, we don't know all the labels. The labels depend on dose, okay, and we always discover new ones, okay. So if it's not in the data, it won't be in the model, okay, and that's a typical life sciences problem. And that's true across all categories of the data we have in the life sciences. Links between drugs, target disease are quantitative and incompletely in many cases characterized. We have subtle differences in compound effects. They can completely block a protein or partially activate and so on. Effects are state dependent. So we have different cell types, different tissues. If you eat something different in the morning, absorption is different, which means your blood plasma level of the drug is different, which means you see different effects and side effects and so on. And overall, the question is, my point here is, the problem is not so much the computer science and the algorithms. The problem is the data. Our data is very conditional. It's very it's very conditional data. It's quantitative in principle, but we don't know all the quantitative data points. So computer science is very powerful, but labeling our data for machine learning is actually the tricky part. That's the bottom line across all the categories of data we have in the life sciences. And that also translates to that problem of having very early stage, as it's called, proxy data. Yeah, we want to predict human effects, but we cannot put every compound into humans. So what do? We have a cell line or very simple model, and we test our compound here and hope that the model extrapolates to the in vivo situation. We can infer something for humans, right? And many of the assays we have work like that. But often those assays are not very predictive. So what is shown here is the activity, so it's called in vitro IC50, but basically the point is the smaller this number, the more active it is. And Cmax is the concentration you have in bloodstream, and the higher your dose, the bigger the compound effect as well. So, and that's the ratio, right? So the more potent your compound, the smaller the number gets, and the higher your dose, the smaller that ratio gets. And then you take the log. So basically, if you go to the left-hand side, you have either a very potent compound or very high dose or both. So you basically have lots of activity, and that's the short answer. Yeah? And you have a test system where the compounds that are here have the biggest effect in a particular liver injury system that's meant to predict if you take that drug, it doesn't do something harmful to your liver, basically. Yeah? And this is the x-axis, so that's the test system. It's called human liver microtissues, but it's a biological test system. And the more active, the more to the left. And clinical reliability is the smaller that number, the more problems you see in the clinic. Okay, and so the x-axis is meant to be predictive for the y-axis, basically. But I think that correlation here that Jack put in here is actually quite optimistic because you don't really see that correlation, right? So the point is that's quite a complex biological model, but the system where you can test data actually has quite low predictivity of the clinical output. It's not a criticism of this particular model, by the way. It's a criticism of biological models and their low predictivity in general. And that's always the situation if we have in vitro models that are meant to predict for the in vivo situation. And this is another example, but I don't go into details. It's called a thermal shift assay. But for virtually every biological system where we don't really understand the mechanisms and we have some type of proxy system, that test system to in vivo extrapolation is very, very difficult. Okay, these are only some of the examples here. And so that means that the machine learning and the life science is actually quite different from machine learning in many other areas. I illustrate that here using example from images and chemistry, biology. So in the image domain, you have an object. Here's a cat in this image. You have a certain 24-bit depth or so. So here it's 1 bit, 0 and 1. So you can represent your image as a matrix. You can use a convolutional network or different other network architecture. So you do certain operations on that particular matrix. Then you have your output bit, and that bit might flip to 1 if there's a cat in the picture, right? And then you have largely unconditional labels. So you can represent your object quite well as a matrix. You can operate with a convolutional network on that matrix. And then you have largely unconditional labels. Largely because the cat can, of course, be partially hidden and so on. But it's not as severe a problem as in biological data, as I illustrated before. So that's actually quite a good integration of the object, the representation, the model, and the label. Okay, it's the same in speech. If you have a waveform, you can actually say that part of the waveform corresponds to the word cat or so, and then you can train a model. Okay, so you can label pretty well. Okay, in chemistry and biology, that's different. So in chemistry, for example, that molecule, you can have different conformations, as it's called, different geometries. You can protonate it, so there's some chemical reactions that can take place. You can have different crystals, yeah. And if you have a different interaction between molecules, some molecules may be solubilized in the body more faster or more slowly. So you have different concentrations in the bloodstream again, and so on. So basically, your object is alive, if you wish. Yeah, it's not one thing. Then you don't know how to represent that particular object. There's many ways to encode that molecule, also related to that particular representation question. Then you have different machine learning methods, and none of them is so neatly integrated, like convolutional networks, for example, operating on a matrix. And then you have conditional labels in the end. Things depend on those. Things depend on endotype and so on. Okay, and so that means that in the life sciences, you have actually much worse integration of the object representation, the model, and the object label. Okay. So of course you can plug things into a model and you get a number, yeah. But the question is, does your representation extrapolate, for example? Yeah, are you able to make out of domain predictions? Yeah, your model, is it actually the number of parameters in your model? Is it actually the right number of parameters? So is your model parameterized properly, and so on? And then the question is, well, how do I assign my labels as well? So that it was meant to illustrate that life science data is quite different from data in other domains, such as image and speech recognition. Also, model validation is often not really considering use cases. That's one of the problems, not only in the life sciences, also more generally, I think. Quite often, if you read a machine learning paper, you have generic model metrics, AUC, for example, average precision, and so on. In many use cases, those generic model metrics, they don't really matter. And in the life sciences, for example, you have typically 2 or 3 different use cases you want to rank, or you have a selection problem or a deselection problem. Yeah, these are 3 typical use cases. There's probably more, but these are 3 typical ones. What you see here is a plot that is plotting recall of objects with a certain label over the relative rank. And so we ask, for example, the computer, out of 1 million molecules, tell me which 100 are most likely to be active. It's a selection problem, for example. So we rank all those molecules, and ideally in the model, the 100 molecules that are truly active are at the top of the list. So we select them for experiments. And in this case, the area under the curve would be basically 1. So it would go up this curve like this. In practice, of course, you don't get that. And in practice, you would get models like here, for example. So model 2 is the orange model, model 1 is this one. And the area under the curve of both models is the same. Yeah, it's exactly the same. But if you have a selection setting, for example, model 2 would outperform model 1 after 10% of the relative ranks in the selection setting. Okay, so the orange model is twice better. Or if I deselect at the other end of the spectrum, then actually model 1 is better. So that's 3.33% here, that's 10%. So here the blue model is actually, if I invert the model, so to speak, is actually 3-fold better. Okay, the area under the curve is the same. And if you basically, if you evaluate your model, you have a performance metric, and you don't have a use case in mind where to use that model, let's say for a selection setting, and I can test 100 compounds, yeah, then what matters is the recall in the top 100 position. That's my use case, because in the experiment, I can test 100 compounds, so that's my use case. Then that's what I need to use to evaluate my model, and not have some generic model metric that doesn't matter in practice, because then both models would be equally good, but in reality, either you're here or somewhere else, but it's not the AUC that matters, okay? So think about how you use your model when you build a machine learning model. That's my point. And especially in chemical space, that's a problem. And the reason is that chemical space is large, 10 to the 60 compounds or so you can make. Typically, if you train a model, you have 10 to the 6 or so data points, sometimes 10 to the 3, but certainly very few. And chemical space is very clustered. So every chemical series, every type of drug basically behaves differently. So that's illustrated here. Every galaxy represents one a system of related compounds basically. And in practice, what you have a training set that you can train a model on a certain type of chemistry. You have a validation set, so you stop training. You have an external test set, and as you can see, the external test set and the training set overlap here. The reason is if you have a database with molecules, also many other areas, of course, and you split your data, even if you don't have, in the strict sense, information leakage, because chemical compounds are clustered, and you put compounds from the same cluster into the training and test set, you still have some kind of information leakage. It's not exactly the same data point, but the training test set split does not represent the future use case of extrapolating to completely novel chemistry. Yeah, so you're lying into your own pocket, so to speak, and this is meant to be represented here. The external test set is not truly external, and but the next project is in completely different areas, different chemical space, different galaxies. And what was actually a model that works on Earth tell me about Venus? Yeah, not necessarily that much. It's a different use context. Yeah, it's different planets. Here it's different galaxies, but it's about the extrapolation, right? And that in practice is a huge problem when it comes to life sciences data, drug discovery. Out of the main prediction, basically, it does not work. It's maybe a bit too black and white, but it's tremendously difficult. And so what I was trying to illustrate here is don't only focus on the model, the use context matters. So if you have a model, you predict something, you have your metric that hopefully matters for the particular use context, but you only optimize the model. In practice, what you have is you have a project context and you have a follow-up. So what do I mean by that? You can train a model for toxicity at a given concentration, let's say 10 micromolar, so a given concentration in a test system. But if you have a project where you dose your drug very differently, so you give much lower dose or higher dose, then it's not the right model in the use context, right? So you basically cannot train a good model without knowing that use context, what matters in the future. But by definition, you don't know that beforehand, right? Because you don't know all future use cases. The other thing you don't know is your model predicts, and now what? Okay, if you spend 20 million on your project and your model says, yeah, 63% likelihood that compound is toxic, you will not scrap the project, right? So the question is, how do you translate model predictions into decision making? Okay, and in practice, so I realized that when I was working at AstraZeneca, heading computational safety group, then the question is, what's your follow-up assay? So what's the experiment you can do tomorrow that's predictive enough to really come to a decision, okay? So don't only build models, always take the project context and the follow-up into account, okay? That really matters. And it matters more for translation to practice to consider that use context than to have an AUC that's 0.01 higher also, okay? That really matters. So model performance and changing processes can be really detached from each other. Of course, the human element is completely left out here, right? So how do you actually change an organization to do that? And the bigger question, of course, is do you operate in a science pull or tech push environment? And they are both completely different in thinking. So science pull means that you have a problem at hand, so you have a project purpose, you want to solve a problem, stratify patients in a given disease, okay? And then you find out, okay, that biomarker could be a good biomarker to use. Gleevec, it's a Novartis anti-cancer compound. You know there's a certain fusion protein. If that is present in the patient, that's the right drug. Okay, so you have a marker. So you generate the right data for that. You use the right method to analyze your data, and then you can use that model. And that's science pull. So you know what you want to solve. Right data, right method, validated in the right way, and you can use your model. But tech push and data push is quite different. Then you have data at hand, and you happily generate data, and then you ask the question, so what do we actually do with it? And then you say, okay, convolutional networks seem to be good, flavor of the day, right? So you apply that method to your data, you evaluate with generic performance metrics, okay? And then you try to find a area to use that model, okay? But actually the steps before were not the right steps, right? You use data that was not generated. It's not the right data for the right purpose, not the right chemicals tested, for example. And then the method, the machine learning method, is not tailored to the data or the use case. The evaluation metric is generic, so also not tailored. And then you're looking for the purpose, right? And that is actually now published, that Drug Discovery Today article. So it's really science pull in many cases, where possible in all cases, I would argue, gives you a better answer to the question, okay? So try to align the things you do. And this is what Koichi said, was at Taijin Pharma, the Japanese pharma company at the time. So he was drawing a comic. I really needed to laugh when I was looking at that. So it's a hypothetical dialogue between DMPKs, metabolism and pharmacokinetics. Can be any experimental scientist in pharma or different areas. And there's a computer scientist. So recently Medchem, so the people who do experiments, has been asking for all the compound data, but it's a pain. I wish to use AI. I use ChatGPT, but I don't know how it works, so I cannot do that myself. AI, I don't quite understand that. Computer scientist, same day. I didn't know there's so much ADME data published. I'm not sure what it means, but numbers are numbers. I just put it into my model. I could write a ton of papers and change drug discovery. So 3 weeks later, so DMPK scientist, I heard from my boss there's an ADME prediction model on GitHub, so I check it out. But the evaluation thresholds are all over the place. Was predicting things that are actually not used in practice. Wasn't really helpful. I probably have to do experiments again. Computer scientist on the same day. The model I released, actually machine learning researchers really like it, but it's not really being used in drug research. I don't know, probably those DMPK scientists don't want to do AI. It could actually reduce the number of experiments and so on. So it's a hypothetical dialogue, yeah, but it really shows that disconnect. And I think what we need to do, all of us, whether we're experimentalists or computer scientists, understand the interface, right? Understand the data, build a model that's fit for purpose, the right data, validate it in the right way, implement it in the right process, yeah? That's where the change then really comes from. Final part, some learnings from big pharma, academia, and startups. So big pharma, so that's from AstraZeneca, an example. What big pharma is doing is called DMTA cycles. You design a compound, you make it, you test it, you analyze it. You do that enough times, and in the end, hopefully you have a compound that goes into the clinic. And what we're trying to do right now is to actually replace the design by a computational generate, evaluate, optimize cycle. Okay. And so what did we do here? So that's an example from AstraZeneca. We were trying to predict if you take a drug, what's actually the concentration you get in the bloodstream over time. Yeah? Because the more compound you have in the bloodstream, the more likely you see effects as well as side effects. And I will not comment on the precise model here. That's not the point. I'm only trying to outline which models, in my opinion, in big pharma actually have a chance to succeed and be used at a given point in time. So this model, so there's some correlations from that model. That model is used to remove the compounds that are really bad. Okay, so that's a deselection problem. You want to remove 20% of the compounds you should not make to save 20% of your resources. So the point where I say that is, it's not really about that correlation. The question is here, high clearance as it's called. So in this, it's not really a quadrant, but in this section of the plot, do I actually select the right compounds that I want to throw away? And that works pretty well, okay? So goes back to the question, which metric should I use? Yeah, and the use case here is removing 20% of the useless compound. And for that, the model is actually good enough. And why do I think, and that's a good model in my opinion, it's where those parameters are as aligned as they can be in reality. Why is that actually the case? And there are a few factors, quite a lot actually. There were pull and push factors. So one is to automate those cycles. Chemists that design new drugs want to consider that late stage parameter very early on. So you need to pull it forward in the pipeline. Predictions are very fast. It's neural network predictions, so a fraction of a second. And you also had push factors. So you had homogeneous data that CROs, contract research organizations in Asia were generating, very homogeneous. You had ML advances, so ChemProp, it's network models, multitask models. And there were biases of the previous, so IVIV means in vitro to in vivo extrapolation models. So the previous models were not good. They were people, some people liked them, but they were not really fit for purpose. Yeah? They covered only part of the predictions they were meant to do. And I think these factors actually contributed to that being a good model at the right point in time. And so the learnings from Big Pharma, in my opinion, so the environment determines what is needed. So my point is, what is needed in the organization? If you try to push out your fancy machine learning models, it's unlikely to succeed. You need to listen to the organization, what they need and would use as well. That's what I mean by the environment, the users. User interfaces are really important. We built quite a lot of 4-class models in the companies. Why that? 2-class models, if you're close to the threshold, or you predict 1 class, you don't necessarily know how close you are to the threshold. Same with 3-class models with the middle class, you also don't know. But if 4 classes, quite often you can predict the trend, yeah, and that can be good enough in many cases. You don't always need quantitative models. Education is key part. So get used to explaining the same thing hundreds and hundreds of times. It's just part of that in a big organization if you want to induce change. And there's quite a big difference between building a model in your Jupyter notebook and rolling this out in an organization that runs across the globe, yeah, where the database runs in Asia and there are users on 3 continents and so on. That's actually something that was really interesting to learn. Academia, so what we did there is we used gene expression data to match drug to disease. So you can say in a disease, a certain gene is upregulated, and so we need a drug that downregulates that gene. That's a simple view. And generating gene expression data is actually quite simple to do. And we did that in many areas. So we selected, for example, compounds based on gene expression data to differentiate stem cells into cardiomyocytes, beating heart muscle cells, basically. It's a very important topic in regenerative medicine, macular degeneration, pancreatic beta cell regeneration for diabetes, and so on. So we want to have compounds that stimulate the growth of stem cells that are present in virtually all body tissues. And it was really fascinating. The computer told me, pick that compound, put it on the stem cell, and after 3 weeks, you actually see a beating cardiomyocyte under the microscope. Yeah, so you create living biology, yeah, by computer algorithms. I found that really fascinating. And that was originally what we started our first company on, Helix, data-driven drug repurposing in rare diseases. So all the algorithms were re-implemented. There's no academic IP involved. It was separately implemented. And that company was based on data-driven drug repurposing using knowledge graphs in the beginning. 2014 was quite early on compared to today. And what are my learnings from that? So Tim, CEO of Helix, came to my office one day and told me he wants to start a company. And I said, sure, let's do that. So it's an encounter as a result of the environment. So I think also here in Abu Dhabi, to grow the life sciences ecosystem, it's very important that those random encounters actually happen as often as we can. There was a real benefit of having the university and startups co-located. The founding team was local. Amadeus as a funder, Amadeus Capital was next door. My PhD students working with the company and so on. It's really synergies, yeah, that you get if you have academia, startups, big pharma in one place. Learning one is don't see how fast change really happens and how fast you think change happens. So I felt that it's quite late in 2014, so a very long time ago, to start a company in the AI drug discovery area. So I said to Tim, yeah, sure, good idea, but isn't it a bit late? That was really one of my comments. It feels ridiculous right now. If you're in academia or work in a fast-moving environment, don't underestimate how slowly the world changes. Yeah, locally it might move very fast, but on the big picture, it takes decades sometimes for the world to change. So try to calibrate your personal perception. Yeah, the world and your own perception might differ a bit. And building a company that's not only tech is safer. If you build something only based on tech and Google wants to do it and puts 500 people on it, they copy what you do in 3 months or whatever it takes. Yeah. So Helix, for example, works with patient groups as well. Yeah, because you need to build that relationship and so on. That takes a while, and that's not just copy paste by some tech people. Yeah. So see if you need a second leg of your company as well. And the other company we built, Farmenable, new chemistry for new targets, very different company, predicting new chemistry for protein-protein interactions, for example, reaction prediction and so on as part of that. And again, local connections were really important. Partnership with Susa Heptares, some startup companies, biotech, or it's no startup, biotech company. And I think that type of company we need to cultivate here in Abu Dhabi as well, so that there's companies that can benefit from local research and can fund it in turn as well. The tech transfer office doesn't get in the way in Cambridge. Basically, you have really freedom to do what you want to do. You can involve it, but if not, then that's also fine. And bootstrapping is very difficult in biotech. We tried that for a year, so not getting external funding, but running it out of revenue and by yourself. Very, very difficult. It doesn't work, basically. And then we raised funding after a year or so, and things moved much, much faster. And the third company, so that's a company that was started in Britain and Germany as well, and now is one leg in Hub 71 as well, is using historical evidence of safety and efficacy for drug discovery from plants, basically. Plant-based medicines. In many countries of the world, plants have been used in health, so you can isolate compounds, and you have some use evidence in humans, right? What matters is clinical phase 2. And if St. John's wort was used across the globe for depression, maybe there's a compound or a mixture of compounds in there that actually works, right? That is the idea. So you have direct in vivo relevance. That company now has compound going into phase 2 later this year as well. What's the learning from the startups in general? So time horizon is different in terms of funding cycles, right? It's an 18 to 24 months funding cycle. At the same time, you need to plan for long term. So it's quite difficult to square the circle sometimes, but you need to address basically 18 months thinking and 10-year horizons at the same time. And that can be actually quite tricky in practice. From the tech side, it's easy in a way. You can build quite fast now in the cloud, right? So in 3 or 6 months, you have a prototype. That's excellent. The key learning is, but that applies to biotech. That's not a revenue generating company. It applies to biotech. Avoid distractions. So work for only the money wherever you can and focus on building assets. The reason is customers always drag you in every direction and want you to do this and that. In the end, you never focus on what you actually want to do. Discover your own drug, basically. That's a biotech statement. Yeah, it's not a tech statement where you want to grow a user base or so. It's different. Tech is different. That's a biotech statement. Yeah, but that's I think what's quite essential there. So to summarize, so current state of AI and drug discovery: check hype and substance and where to position yourself there. Data and model process disconnect. So if you build a model, make sure it's the right data for the right purpose, and that's aligned with the process. And different environments, big pharma, academia, startups require very different approaches. 2 slides here on drug discovery in the UAE, and then I'm done. So we are starting a drug discovery, biotech, and beyond network, and we have some experimental facilities which might be of interest for you if you're interfacing with drug discovery as well. So the first one is this UAE drug discovery, biotech, and beyond network, and you can go to that website in case you're interested, genetic.ae. So we are trying to facilitate interactions between scientists in drug discovery and all its interfaces. Also includes digital health, for example, health apps, bioinformatics, AI and life sciences, and so on. So it's now a few hundred people or so that are on the network. We had our first event in June, was 400 or so people registered. If you know of a WhatsApp group and a website and so on. And later this year, we want to have another meeting here in Abu Dhabi as well, probably at Khalifa University and then around the country, in order to bring people together, scientists together, and then to work towards translation to stimulate that ecosystem. So if you're interested, everyone is very welcome to join us. Just go to the website here, genetic.ae. And the other thing is, so we actually have quite fancy facilities coming up at the university, Khalifa University. Cryo-EM, structural biology. So if you need protein structures, let us know. It's really powerful machines. So we can use them for academic collaborations as well as fee for service. And we have a large animal house coming up as well. It's the largest here in the Middle East, where we can do trials, studies of compounds and so on in animals as well. 70% mice, 30% rats. And that's very flexible too. So we can do academic studies as well as fee for service or work with companies as well. So just let me know in case you're interested in working together. Yeah, I think that was everything. Get in touch if you wish, and thanks for listening.
We'll open the floor to a couple of questions.
Hello, I came from Dubai and I'm very happy I did it. I abandoned WHX, so it's worth it. Thank you very much for the talk. My name is Rasham Salam. I'm an immunologist, scientist, and have a consultancy in immunology in Dubai. I work a lot in linking academia to startups. I used to do that in Singapore, and now I'm trying to link Southeast Asia with the MENA, specifically UAE. For the facilities, because I get contacted by some startups who wants to expand to UAE scientifically based, not commercialization yet. So they want to build. How can we build that? What do you advise? They look to rent wet labs, and sometimes they have funding, sometimes they can be part of grants. I didn't see anything about the grants as well. So they look for equity-free support. Is it whether in kind or funding? What do you advise? What do you think? How we can build it as well? Thank you.
I think so. Things are developing in the right direction here. There's actually there's not that much wet lab space, for example, around in Abu Dhabi right now, but in Masdar, as well as Khalifa University on the Sun campus, it's the one closer to the airport. There's now labs being established, wet lab and microscopy and so on. There's now a few things coming up. Bring them in touch and we can see what we can do. Yeah, and then we can see how to mesh that and so on.
Okay, another thing is I'm very fond of mentorship. Students must have real life experience as mentorship, not only to help them to publish, is to face reality that as the job market after having the fancy PhD. How do you think also this network can help these students? I don't know if there are students here, to help them to realize that it's not only the supervisor will support you, you need to learn in the hard way. Because this is also something, I think, as building in academia/startup, they need to know that, right?
Yeah. That is so, and that's I was always trying to cultivate that in my environment as well, to bring students in touch with people from a company to give feedback on what they are doing, for example, because it not just does it work for publication feedback, but does it work on reality feedback, and to bring people in touch with each other, and I'm very happy to do that from my side, and that is a culture I think that we should build as well, It doesn't need to be a formal mentor, it can be, because then that person has dedicated time, but bringing people in touch with each other as much as you can, people you can learn from or the student can learn from. I think that is key. I think so as well. And ideally in our area, computational life sciences, at least from those two areas, academic and companies, for example, to have a matrix of feedback, basically. I think that's a good idea.
Thank you so much, Andreas. Great talk. I'm Halima Naqbi, an assistant professor from Khalifa University. And my question to you is, it's because you've really, distinguished between drug discovery, both in academia as well as in big pharma, right? And my question is, how should we think about the strategy or the entry point when we're looking into, drug discoveries, perhaps mystery targets that we want to later on patent on? What I noticed that mostly in research and academia, we start from a kind of a disease-centric strategy. We start from the disease pathology, we understand what's going on in the biological system, and from that we develop targets, or we predict that those targets, and when we use AI and ML and so on. But how does perhaps starting from a compound itself, repurposing that for major objectives differ, and which strategies from your point of view or from your experience do you think is the most like the straightforward one, the most straightforward one? Yeah.
Yeah, and that is what you just described. That's actually very valuable, I think, what academia is doing. Pharma is in dearth and searching for new targets. Yeah, how do we should we address that disease in across a wide range of diseases? Yeah, and if academia comes up with a new target from omics data analysis and some type of in vivo proof of concept, yeah, in animal model, for example, I think that would be very valuable. Yeah, so you have an in vivo that you can cure disease using a small molecule, which can be an existing drug, by the way. Yeah, so I don't think academia would need to come up with a completely new compound and IP and so on, proof of concept, and then partnering, yeah, with that company, with a pharma company. Of course, if you can do this step in-house, but very few universities would be able to come up with a novel compound as well. Yeah, but I think that's actually what we want to work towards at Khalifa University as well, to build the whole pipeline. There's now the Institute of Health, for example, coming up in the context of the university, right? And to actually have an integrated drug discovery environment, that's actually something that I'm also involved in working towards, to actually do the full thing. Yeah? But coming up with a target, a new biological mechanism to treat a disease, and having in vivo proof of concept, for example, in our new animal facility, I think that is actually an excellent idea, yeah? Because then you know something that might really work in humans as well. Yeah, so that type of sharing of actions, yeah, between academia, discovering the novel stuff, pushing it far enough, and then out licensing, partnering. That's actually, if that works and we make it work here, I think that's a very good starting point, actually.
Yeah, thank you.
Thank you very much. It's nice to see so many people from Khalifa University here. I'm George Zubedi from the Mathematics Department. So my question is actually, what do you think is the role from a more theoretical point of view, like causal inference in this drug discovery? Do you think it is already integrated in the production, in the pipeline, or do you think that there's a long way to go?
Yeah, causal inference is, I think that is the direction we need to move into with omics data. Yeah, to have some directionality, to go from correlation to causality, because quite often in biological systems, you don't know is actually, do you see a consequence of the disease or a cause of the disease, right? So if you're able to use causal inference methods on omics data, for example, we try to do that as well. It's very tricky, to be honest, in my experience, but I think that is to have causality, quantitative effects in there. Yeah, I think that is a key direction. Another one is pairing like in prior, you would say in Bayesian methods, yeah, in knowledge with data, yeah, to combine those two to reduce the hypothesis space. I think that's one of the other big avenues to go into. Not just needle in a haystack, we generate high-dimensional data and fish in it, yeah, but we combine information, network information, with data. I think that's one of the other big avenues, for example. Yeah.
Thank you very much.
Hi, thank you, Doctor. My name is Mira. I'm both a student and at Khalifa University, and I work at DOH. I just wanted to ask, you've talked about the gap of clinical trials when it comes to AI drug discovery. Can you elaborate on what's the lag or pullback when it comes to the clinical trials and investors investing in them?
So, I mean, AI and drug discovery is quite a broad spectrum, right? So where data can help, and that relates quite a lot to genomics data as well, is to stratify patients where I know enough that data pushes me over the line. So what do I mean by that? If I'm able to stratify patients cleanly enough in order to have a cohort that responds sufficiently to drug treatment, yeah, that is, for example, an area that's a success story. Like I think I mentioned it in cancer, you go from 3 to 8% across different organ types and so on in cancer success rates. Yeah, so I think that is, for example, a success story. The question is quite often, if hypothesis-free omics data, you don't see enough in the data to really use it. Yeah, so that can be tricky. In some diseases, you have very high dimensional data, small cohorts, especially if you go into subgroups of your data. Yeah, your cohorts can get very small. Also, UK Biobank data also, yeah. You don't have enough data. It sounds good, but you always look at subgroups at some stage, and the subgroups can be quite small. And so I think where we understand enough, that data allows us to make the right decision, for example, in patient stratification. Yeah, biomarker, so companion diagnostics is called, right? So you have a biomarker as well as a treatment, a therapy. Yeah? And we need to identify where we know enough. What pharma companies did the last few years as well. That's why you have quite a lot of contraction over the last 6 or 8 years. Pharma companies decided, you know what, if you just try to do everything, it doesn't work. Let's focus on the diseases we understand enough, where we have the right compound, we know there's target engagement and so on, to focus in areas where we know enough, so we are likely to be successful. Yeah, and that's only a minority. I think it's an illusion to think that Demis Hassabis, I think, said that in 10 years we cure all diseases. Yeah, no, will not happen. Yeah, but if we cure the 20% where we have enough understanding and data, hey, that's pretty big achievement already. Okay, and it's better, I think, to put resources on that, yeah, where we understand enough, yeah, to have clinical proof of concept early enough to be successful then. Yeah.
Thank you.
Hi, doctor. Thanks for the talk. I had a question. So for the past year and a half or two, ever since ChatGPT came out, we've heard all these headlines about how AI is going to revolutionize healthcare in general, but pharma too, and how AI is going to help with this mutation prediction and gene expression modeling. And how much of that do you think is pure hype versus actual kind of breakthroughs?
Yeah, that's exactly what we are trying to do, to put things into practice. There are a few areas where language models also in our hands are very, very useful. You have information in heterogeneous data sources, is more or less what ChatGPT does as well. In a big pharma company, you also have 200 databases, written reports. Yeah, no one knows what we know about that compound. Yeah, to compile that, pretty useful. Yeah, to have your language model, for example. Summarization of documents, information extraction. So in one of the companies, Pangaea Bio, we also try to extract information from literature about plants and compounds and uses and so on. You get recall precision 0.8 or somewhere there. Yeah, it's pretty useful. Yeah, it's good enough because you can actually then map it to some dictionaries you have. So you reduce the number of false positives. It's useful. It's useful enough. For decision making, I think it's very, very tricky to use this type of models, yeah, because you need quantitatively weigh evidence in absence of complete understanding. Yeah, so you don't have complete information. You don't. As a medical doctor, in this particular tissue, in this particular patient, you have an overexpression of that pathway or so. Yeah, you have latent knowledge that is difficult to make explicit, and the model doesn't know that. Yeah, so it doesn't know pieces of the puzzle basically. And in those areas, so quantitative weighing of evidence in absence of complete information, very, very tricky. Very, very tricky. Yeah, you need to see where to apply which model. That's more or less exactly my finding as well. Yeah, to align the model, the data, the use case to make sure it works. Some areas, yeah, but not everywhere where large language models are currently proposed. Yeah, that will really work out. I mean, certainly not.
Thank you, everyone. Any further questions can be directed to the speaker's email or in our networking event. One drug we're going to all discover is insulin. I hope you enjoy the refreshments.
Brilliant. Thanks, everyone.